Now let us see this 505J in a little bit of detail, that is how it differs from the other 505 applications. Now it contains data submitted to FDA CDER OGD division reviews and approves generic drug product. Once approved, the applicant may manufacture and market generic drug product to provide safe, effective, and low-cost alternative to American public. All approved products, both innovator and generic, are listed in FDA's Orange Book. So the ANDA applicant also has to submit the data, that is the quality data, the BE data, to the agency for review. And once the ANDA is approved, what happens is since it's a generic drug. Why it is a low cost alternative? Because the costly animal studies, the costly clinical studies are not required to be done by the generic. It is already proven that that API or that drug is safe and effective. Hence, the animal studies and the clinical studies need not be repeated again by the generic applicant. They just have to do the quality part of it and the B part of it and give it to the agency for approval. Hence, their research cost is much low. The development cost is much low as compared to an NDA applicant. Hence, their the final cost of the product that comes into the market is also low. So even if it is a copycat, that is, even if it is the same quality as an NDA, that is, as an innovator product, it is a low cost alternative. Means if the for example, if the RLD product is sold at say $1,000, the generic can be sold as low as $20 or $25. That is the cost difference that comes into the picture when generics enter the market. Now, generic drug applications, termed abbreviated, as they are not required to include preclinical and clinical data to establish safety and effectiveness. We have seen that because it is already established by the innovator drug applicant. Generic applicants must demonstrate that their product is bioequivalent, that is, performs in the same manner as innovator drug in the human body. Generic version must deliver same amount of active ingredients into the patient's bloodstream in the same amount of time as the innovator drug. So this line, the last line, is what is meant by bioequivalent. In layman terms, that is what is bioequivalent. So it has to perform in the same manner as the innovator drug in the patient's body, in the patient's bloodstream. So that is what is required to be demonstrated. Means with respect to clinical studies, that is with respect to human studies, this is the only study that the generic applicant has to do. So in terms of data requirement, if we compare the data requirements of an NDA, that is a new drug application, or the innovator drug application, and an ANDA, the difference is only in the last 3 lines. That is the animal studies, the clinical studies, and the bioavailability studies, which are required for the innovator drug, are not required for the generic drug because it is already established. What is required instead is the bioequivalence part. All the other things are the same. Now, one important aspect here is the labeling of the generic also. The generic applicant, that is the ANDA applicant, need not develop. It has to be an exact copy of the innovator